Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 5 for May 2021. I will do this paper into videos and I'll put the link to the part 2 video below the description box. Let us begin. Question 1 says, aerobic respiration involves 4 stages, or these are 4 main stages. We have glycolysis that takes place in the cytoplasm, the link reactions in the Krebs cycle that take place in the matrix, and oxidative phosphorylation that takes place in the Christie. They say one product of all 3 stages shown in the table is ATP, adenosine triphosphate. They want you to complete the table to show one other product of each stage. So here we can see glycolysis, of course. Along ATP, there is pyruvate that can be produced as well as reduced NAD. In the Krebs cycle, there is carbon dioxide, reduced FAD, as well as reduced NAD. And in oxidative phosphorylation, water is produced as a final product. There is the FAD and NAD. These are not reduced. Moving on, an incomplete equation for the aerobic respiration of a substrate is shown. This is the equation. They say complete the equation by inserting the numbers on the dotted line to balance it. So imagine this was not there and that was not there. Usually, if an organic compound is given to you and is kind of a combustion reaction, you begin by balancing the carbons. So since we have 57 carbons here, you put a 57 here, since all the carbons in the reactant ends up as carbon dioxide. So that should be 57. The next step should be balancing hydrogen. However, hydrogen has been balanced for us. If it had not been balanced for us, we could begin by looking at this. It's 104. And this side we have a 2. So 104 divided by 2 creates a 52. So 52 times that, it creates the 104, which is similar to what we have here. And finally, you go to the oxygens. We see this side has, of course, we already have 57 here. So 57 times 2 plus the 52, it's going to give you an 80 on this side. So we can see this is 80 oxygen molecules, as we can see. So this is well balanced. Moving on. Here they say 1 molecule of glucose contains 2867.48 kilojoules of energy. And they say 1 molecule of glucose generates a maximum of 38 molecules of ATP. So that means if glucose contains that amount of energy, yet it produces 38 molecules of ATP through aerobic respiration, that can be quantified. And then they go on to say a molecule of ATP contains 30.51 kilojoules of usable energy, meaning if they are 38, the total usable energy should be 38 times 30.51 kilojoules. So here they say usable energy is available for chemical reactions in a cell. They say which reaction releases energy from ATP. Energy from ATP is released by hydrolysis reactions, as I'm going to show you here. An ATP molecule is hydrolyzed. This is hydrolysis to produce ADP. This is D plus an inorganic phosphate. And in this process, energy is given off or energy is released in cellular reactions. So the answer here should be a C. Moving on. They say calculate the maximum percentage of energy in a glucose molecule that can be converted into usable energy in an ATP molecule. So here we can see, remember we say 38 molecules of ATP are going to be released, and a molecule of ATP has this amount of usable energy. So total amount of energy contained by the 38 molecules should be that times that. So it means here when I'm finding that should be 30.51 times 38 divide by the total amount of energy contained in a glucose molecule times 100, which gives me 40.43%. So my answer is 40.43%. Moving on, here they say, describe what happens to the energy that is not converted to usable energy in a muscle cell. This energy can be lost as heat, which is used to raise the temperature of the body. Remember, this is a 2-mark question and they're asking you to describe. So you have to talk about how this heat is used. Lost as heat in order for the body's temperature to be raised. Part D says, describe how oxygen is involved in the production of ATP on the Christie in the mitochondria. During oxidative phosphorylation, oxygen acts as the final electron acceptor as well as the final proton acceptor in order to produce water. So here I said, since electrons are released to the electron transport chain, oxygen is the final electron acceptor in the electron transport chain. It also accepts protons, or it gets reduced to form water as the final product of oxidative phosphorylation. So this brings us to the end of question 1. Let us continue to question 2. Question 2. The nervous system of an organism enables it to respond to a stimulus. A reflex action is a rapid involuntary movement in response to a stimulus. The response to a pinprick in a finger is an example of a reflex action. They say, which component of the nervous system continues a reflex act immediately after the receptor has been stimulated by the stimulus? This is a receptor, and this is going to cause the generation of an impulse, which is going to be carried by the sensory neuron towards the central nervous system. So the answer here should be a D. The next part says, which pathway shows a reflex act? So here I chose C, which is the receptor. Then it's carried through the sensory neuron to the central nervous system, in this case the spinal cord, and then through the motor neuron back to the muscle. So it should be a C here. The others don't make sense. So let's continue. Here they say Pacinian corpuscles are pressure receptors found in the skin of the fingertip. The effect of different pressure applied to the fingertip or on electrode potentials across the axon membrane of the neuron of Pacinian corpuscle was investigated. They say the diagram shows the structure of this structure here, and they say the location of the electrode. So electrodes, you can see there is electrode X and electrode Y used to measure the axon membrane potentials. So they say the table shows the results of this investigation. So we can see there is if the pressure applied to the fingerprint is none, it means the membrane potential was negative 70 millivolts, meaning there is no depolarization. As well as here, the membrane potential at electrode Y for X and Y, there is no depolarization. So when they apply a lower pressure, we see there is negative 60. So it means there is less depolarization, but here there is no depolarization. Medium caused a positive 20. There is depolarization. Here we see positive 40. There is depolarization as well. A higher caused positive 40 for both. There is depolarization. So here we can see the all or nothing law being applied. So the first question asks us to describe how the resting potential of negative 70 millivolts is maintained in the axon when no pressure is applied. Here we have to talk about the sodium-potassium pump as well as the movement of ions across the membrane. So I said, at rest, the sodium-potassium pump removes sodium ions from the membrane as potassium ions are transported into the membrane. So basically, there is going to be pumping of sodium ions out as potassium ions are brought inside the membrane. And then the sodium ion channels are closed, so the sodium ions cannot re-enter the membrane once removed. So on the other hand, the membrane is permeable to potassium. So imagine a situation like this, whereby sodium can be pumped outwards, like that out, but it cannot come back in, so that is out. However, potassium is pumped in, but this potassium has a way of going back outwards based on the concentration gradient. So it means there is a higher chance that the outside part of the membrane is going to maintain a more positive charge in comparison to the inside of the membrane. So here, continuing, I said, on the other hand, the membrane is permeable to potassium ions because potassium ion channels are open. They should be They are open. So the potassium ions can freely move out of the membrane, and there is a higher concentration of sodium ions outside and high potassium inside. So in this case, it means there is going to be, at resting potential, the membrane is going to have a more negative charge inside in comparison to the outside. Here they suggest how a change in pressure at the Pacinian corpuscle causes an action potential in the axon. Remember, change in pressure is going to activate, maybe some molecules or membranes are going to change their shape, and that is going to cause an influx of sodium ions. As the shape of the membrane changes, ions are going to be able to go inside. So I said, application of pressure causes deformation of the cell membrane in the Pacinian corpuscle, and this causes the influx of sodium ions into the membrane, and depolarization can occur. The reason as to why I chose this approach is, let us go back here. The higher the applied pressure was, as we go moving this side, there was more depolarization with the increase in the pressure being applied until the depolarization, or until the membrane potential reached 40 millivolts. After 40 millivolts, no matter the pressure, you can see here, after 40 millivolts, no matter the pressure being applied, there was no change in the membrane potential. So it means the higher the pressure applied, the more the ions were able to move across the membrane, and therefore the more depolarization occurred due to the movement of sodium ions into the membrane. So moving back, I said, this causes the influx of sodium ions into the membrane, and depolarization can occur. If the depolarization is above the threshold, then an action potential is generated in the axon. So that's it. Let's continue. Here they say, explain why the membrane potential at electrode Y was the same when medium or high pressure was applied to the fingertip. Here we have to think about the all or nothing law. Once a specific potential is reached, everything is going to be, you cannot cause a more depolarization or more change once the threshold has been reached. It creates the same action potential. So I said both medium and high pressures cause depolarization above the threshold. This is the negative 55 millivolts, and based on the all or nothing law, both generated an action potential. It has the same magnitude. And again, remember the keywords are those underlining and putting some marks on. It means it has to be above the threshold, and then based on the all or nothing law. Once this negative 50 millivolts is reached, the all or nothing law is going to be followed, and therefore an action potential must be generated. So this brings us to the end of question 2. Let us continue to question 3. Question 3. Many neurons in the human nervous system are myelinated. They say a myelinated axon conducts impulses faster than a non-myelinated axon of the same diameter. They want you to explain this difference. So the key thing about myelin is it acts as an electrical insulator, and wherever it is positioned, the action potentials will not occur in that area. So it means the places where the myelin is not positioned, that's where the action potentials are going to occur. So here I say the myelin sheath is an electrical insulator around the axon. The action potential therefore only occurs at the nodes of Ranvier as the nerve impulse jumps from one node to the next. If this is an axon like that, we are going to have those, the myelin here like that. And this area here is what you call the nodes of Ranvier. So that is a node of Ranvier. It means if there is no myelin, only action potentials will occur at the nodes of Ranvier. And so it will look as if they are skipping from one node of Ranvier to the next one. Let me show this side as well. As if it's skipping from here, we'll see one action potential here, and then there, nothing will be occurring here. However, if we compare a non-myelinated neuron, the action potential has to occur all across the neuron, and that is going to be time-consuming. So here, down here, I put extra explanation. I say, for myelinated neurons, depolarization only occurs at the nodes of Ranvier, creating action potentials only at that point or at those points, and the action potential jumps from one node of Ranvier to the next. For non-myelinated, depolarization should occur across all the axon or the whole axon, taking longer. So next part, they say, polyneuropathy is a disorder that damages the myelin sheaths of neurons throughout the body. One symptom is muscle weakness. Muscle weakness is the reduced strength in one or more muscles. Suggest how damage the myelin sheath of neurons can lead to muscle weakness. Remember, we talked about the myelin sheath. If it's damaged, it means the impulses are going to travel slower, and that means the muscles will not be able to respond as they should be. So I said, when the myelin sheath is damaged, the ability of the nerve impulse to move along the axon is going to be impaired, or it's going to be slower. So fewer impulses will therefore reach the muscles, and less muscle stimulation will occur. This will lead to muscle weakness. So if there is less muscle stimulation, there will be less muscle contraction, and therefore they will not be able to take part in some activities that are required. So let us continue. Here they say dementia is a condition associated with the ongoing decline of brain functioning. There are many types of dementia. They say the relationship between myelin in the brain tissue and types of dementia has been investigated. They say the mean quantity of myelin in samples of brain tissue from groups of people with types of dementia and from a control group was measured. So the table shows the results of this investigation. So we can see here they say the control group without dementia, the mean quantity of myelin in a brain tissue is higher in comparison to all the other groups. Here we can see the standard deviation, of course, plus or minus. So if we are given the standard deviation, then we have to see if there is overlap in the data or not. So patients with vascular dementia, we can see they have the least quantity of myelin. Those that have Alzheimer's dementia, they are in position 3. And the patients with Lewy body dementia, these are closer to the control group. So the value is a little bit higher in comparison to the others. So I tried to write some data here in blue to show the standard deviation so that we can see if 52 lies between the higher values 55.2 and 49. Here are 25, 31.9 if you add 5.9, and if you subtract it will become 19.1. 32, adding a 4.1 you get 36.1, and subtracting a 4.1 I got 28.9. At a 42, adding a 5 I got 47, and subtracting 5 I got 37. So this gave me the highest value and the lowest value so that I can see if there is overlap in the data as I try to answer the questions that follow. So here they say, a student concluded that there was a relationship between the quantity of myelin in the brain of a person and whether or not they had dementia. They want you to comment on the validity of this conclusion. So based on the data I said, all samples from dementia patients had lower quantities of myelin in their brain tissue than the control group. So we could use this to see why this conclusion could be made, because those that the control group had a way higher brain tissue sample than those who had dementia, basically. So I went on to say that the results, however, do not show a relationship between the amount of myelin present and the type of dementia. Based on the standard deviation, there is no overlap for any type of dementia, so the results may be valid. Based on this, you can see I tried to put the highest and the lowest, and there was no overlap, so I said, because of this, the result from this experiment could be valid or could be believable because values for different groups did not overlap. So this brings us to the end of question 3. Let us continue to question 4. Question 4, that kidney is involved in the process of homeostasis in a mammal. Antidiuretic hormone is required for homeostasis. How many of these parts of the kidney respond to ADH? This is the hormone they talked about here. So they say the first one is the Bowman's capsule. That is wrong. Collecting duct, that is correct. The loop of Henle is wrong, and then the proximal tubules are wrong. So it's only one among the 4. That is why my answer here was A. Moving on, they say the electron micrograph shows a transverse section of part of a proximal tubule. This is what they're talking about. They say name the process by which the fluid moves from the plasma into the renal tubule. This is by ultrafiltration, so that is why I put it here, as this fluid leaves the blood vessels, the tiny blood vessels, to go into the space surrounding that. So the part 2 says, name the structure labeled S. These are tiny, tiny projections. You can see those are microvilli, and I wrote it here. Let's continue to the next page. Here they say the student compared the left renal veins of 2 individuals. The diameter of the left renal vein of one individual was 4.71 millimeters. Calculate the cross-section area of this vein. Since I am given the diameter, I'm going to use this to find the radius. We know that radius is diameter divided by 2, which is 4.71, divided by 2, giving me 2.355 millimeters. You could also round it off to 2.36 millimeters. Then I use this to find the area. I said area is pi r squared because, of course, this is just cross-section area. So area is pi r squared, which is going to be pi times 2.355 squared, which is 17.4. And my answer was 17.42 millimeters squared. The next part says the cross-section area of the left renal vein of the second individual was 0.194 centimeters squared. They want you to calculate the difference in the cross-sectional area in the left renal vein of these 2 individuals. So I needed to convert this and that to be in similar units. So I used this and converted it to millimeters squared. The key thing is, if 1 centimeter is 10 millimeters, 1 centimeter squared should be 100 millimeters squared. So it means this times 100 will give me the millimeters squared I'm looking for. So I got 19.4 millimeters squared. This is for conversion of this into units similar to that. So after getting that, I had to get a difference in cross-sectional area, which is 19.4, which is this one here, minus 17.42, giving me a 1.98 millimeters squared, and that is what I got here. So the answer could also be 1.97, depending on how much you rounded off here. You could get that as well. So moving on. Here they say The table shows the concentration of glucose and urea in the blood plasma, glomerular filtrate, and the urine of an individual. So we have the substance, glucose. We can see the concentration in the blood is 1.5. The concentration in the glomerular filtrate is 1.5. This means that all the glucose has been removed. And then the concentration in the urine is 0. That means all the glucose has been reabsorbed. Now for urea, we can see in the blood is 0.3, in the glomerular filtrate is 0.3, and in the urine is 21 gram per decimeter cube. This means the concentration is higher because a lot of water has been reabsorbed, so we can see it's a higher concentration. Here the question asks us to explain the concentration of glucose and urea in the urine. Use the information in the table to support your answer. So here I said, during ultrafiltration, all the glucose and urea pass out of the plasma. The proximal tubule, so this part here is for why the concentrations in the blood are exactly the same concentration as we can see in the glomerular filtrate. That is for this part. Then the next part I say, the proximal tubule selectively reabsorbs 100% glucose using protein channels present along the membranes, and therefore no glucose will be present in the collective ducts. This active reabsorption or selective reabsorption uses energy from ATP sometimes in order to ensure that all the glucose has been pushed to the other side or back into the tissues nearby. Then lastly, I said because there is water reabsorption and no reabsorption of urea, the urine produced will be 70 times more concentrated. If you can compare that to that, it's like way high. So I said 70 times more concentrated than the filtrate basically. So this was a 3-mark question. You could just write like I did here and you get the marks. Remember, this is that, and that were awarded. So next here they say an individual produced 0.83 centimeters cubed of urine each minute. Calculate the volume of urine produced by this individual in 24 hours. You assume the urine production is at a constant rate and give your answer in decimeters cubed to two significant figures. So here what I did is I had to calculate how many minutes do we have in 24 hours. It will be 24 times 60, which is 1440 minutes. And then you remember here we had, so the production, if this is per minute, it means that times this time will give us how much urine can be produced in the 24 hours. So 0.83 times 1440 gives us that centimeters cubed per hour. Now to convert this to decimeters cubed, I had to divide by 1,000. So divide that by 1,000, we get 1.1952, which is approximated to 1.2. Because they say two significant figures, it will be 1.2 decimeters cubed. And that will be our answer. So this brings us to the end of question 4, as well as the end to this video. Thank you for being with us. Please do not forget to subscribe. See you in the next video. Bye-bye.